Thank you for the kind introduction. Thank you all for being here. It's an honor to be behind this lovely desk with a sink in it. I've never stood behind and presented from a desk with a sink in it before. So my book is I Contain Multitudes. It is out today. And in it, I talk about this hidden world that influences all of our lives. But to talk about that world, I want to first take you back in time. I want to start 350 years ago and take you to the Dutch town of Delft, where a man called Anthony van Leeuwenhoek is about to change the world. I know there are a few Dutch speakers in the room, so I will forever apologize for the way I pronounce his name for the rest of this talk. He was not a scholar, he was not a scientist, he was a draper and a civic servant. But he had two qualities that are incredibly important for our story. He had an insatiable curiosity about the world around him, and he was the maker of the most powerful microscopes of the day. He made them himself. He ground these impossibly perfect lenses and sandwiched them between two brass plates, creating an object that looked a bit like a glorified door hinge, but was in fact better at magnifying the world around him 10 times better than anything else available at the time. And Leeuwenhoek turned these microscopes onto everything at the time, onto bits of animals and plants, into water. Water that collected in an urn next to his house, water from the canals and lakes near where he lived. And wherever he looked, he found a multitude of living things. He found tiny organisms in extreme numbers, moving about very prettily and completely below the threshold of his perception. And these were bacteria. Microbes, microscopic organisms, and Leeuwenhoek had just become the first person to see them with his eyes, to know that they exist. What a momentous point of history that was. He also realized that they existed on his own body. He took samples of dental plaque from his teeth and looked at that under his microscopes too. And again, he saw multitudes of living things. And he realized that there were more creatures living within his mouth than there were people living in the entirety of the Netherlands. Was he disgusted by this? Was he revolted, as many people today would be? No, he wasn't. He was actually very excited. He was amazed and enthralled to learn about the life that shared his body. The view we have today of microbes is very different. We think of them as agents of disease and of dirt and filth. And that's because a few centuries after Leeuwenhoek made his discoveries, in quick succession, it became clear that many of the most infamous plagues of humanity were in fact caused by bacteria, by infectious germs. Everything from tuberculosis to plague, to cholera, to leprosy, gonorrhea, syphilis, all of these terrible diseases were the work of microbes. And so they were forever billed as things that killed us, things that we needed to destroy. If we saw them, it was only because of the symptoms and the lesions and the pustules that they caused on our bodies. And that framing very much continues to this day. Headlines like this abound in the papers. Every time someone takes a swab of a random surface, be it a desk or a microphone or a piece of clothing, and finds bacteria in there, the implication is always we've done a poor job of cleaning ourselves, that we are wallowing in our own filth and are imminently going to die. The Daily Mail will tell us that about anything, but in this particular case, it's talking about microbes. And this reputation is deeply unfair because only a small minority of microbes are actually responsible for disease, maybe 100 kinds or so. The vast majority are actually benign or beneficial to us. And they are, in fact, the lords of the planet. They run the show. We are just a footnote in their world, and they've been here for the longest time. So if we consider the length of this very famous desk as the entire history of life on Earth, so that life originated on here, and that we sitting here in the present moment are on this edge, then humans arose here. The dinosaurs reigned from about here to about here. And multicellular life, so all the things that you can see with your own eyes, animals and plants and so on, arose here, where this pen is. For the rest of the history of life on Earth, till its origins roughly here-ish, it was entirely microbial. You would not have to be able to see it with your naked eyes, which would have been fine because eyes evolved here. So, we are but the icing on a grand microbial cake. When animals evolved, we did so in a microbial world, in a world that was already dominated by and run by bacteria and other microbes. And we evolved to live with them, to exploit them, and to crucially depend upon them. To this date, each and every animal on the planet, every hawk and hummingbird, every rhino and sperm whale and stingray is a vast community of teeming multitudes, a zoo in our own right, and a multi-species collective. Even though I seem like I'm standing here alone, one person behind this desk, I am in fact multitudes, as the American poet Walt Whitman wrote in Song of Myself. Okay. Every animal is like this. So this is a sponge. This is among the simplest of animals. It is just a couple of layers of cells sandwiching a jelly-like filling. But it too contains multitudes. It too is home to a thriving microbiome, a community of microscopic organisms. Every ant lives in a colony that can number in its millions, but each ant is a colony in itself. A polar bear that's trundling solo through the Arctic is in fact completely surrounded. Bar-headed geese fly microbes over the top of the Himalayas. Sperm whales dive them down into the deepest oceans. And when these people set foot on the moon, they were also taking giant steps for microbe kind. These little organisms that share our bodies are not just stowaways. They aren't just hitchhikers. They are, in fact, really important parts of our lives. And in many species, they bestow their hosts with incredible superpowers. This adorable creature is the Hawaiian bobtail squid. It will fit in my hand. It has kaleidoscopic color-changing skin, like most squid. It also has glowing bacteria in its undersides. And the light they produce shines down below the squid and perfectly matches the moonlight that shines on top of it, so that any predator watching from below cannot make out this animal's silhouette. Thanks to its bacteria, it casts no shadow and it is invisible to the things that would hunt it. These tiny white worms kill insects by vomiting toxic bacteria into their bodies. This bird is the hoopoe. It paints its eggs in bacteria that produce antibiotics, bacteria that kill other microbes, and thus prevent the chicks from coming down with lethal infections. This is a spotted hyena. It writes its autobiography with microbial scents. It has bacteria in a gland on its backside, and the chemicals that they produce correlate with the hyena's species, age, sex, and social status. They reveal who this animal is. And this is a flatworm called Paracatenula. Its entire body is largely filled up with microbes, blue in that image. If any of those remain, this worm can regenerate its entire lost body. If you cut it in half, it will regrow, thanks to its symbiotic multitudes. We humans also rely on microbes for our development and our health. They help to digest our food. They help to steer the growth of our organs. They help to prevent disease. They may even influence our behavior. I've been writing about this field of science for actually over a decade now. And there are very few areas of biology that intrigue me more, because I have always been fascinated by the natural world. I went to zoos. This is an image of me with a kid on my back. I watched David Attenborough documentaries. I'm sure many of you have as well. I read wildlife books. But now, thanks to my understanding of the microbiome, I know that every one of the residents and visitors in the zoos I visit is a zoo in its own right. Everything I can see, every animal, every plant, is profoundly influenced by things I cannot see. And if we don't understand them, if we don't understand the microbes that live with us, we don't understand ourselves. We are like looking at our biology through a keyhole. And through writing I Contain Multitudes, I wanted to throw the door wide open. I wanted to expose people to this grander view of life that comes from understanding the microbial world. Because when you do, even the most familiar and mundane things can become newly amazing and wondrous. So here are some examples. Breastfeeding seems like a way for a mother to nourish a baby. But in fact, about 10% of breast milk contains these sugars, human milk oligosaccharides or HMOs, complex sugars that the babies are incapable of digesting. Those sugars are there to nourish the baby's first microbes. And particularly some strains like this one, Bifidobacterium longum infantis, or let's just call it B. infantis for short, that have evolved to specifically digest those sugars with incredible efficiency. And when they do so, they also nourish the baby's gut cells. They seal up the lining of the infant's gut, and they quench inflammation. So a breastfeeding mother isn't just nourishing a baby, not just nourishing an infant, but infantis as well. She is building an entire ecosystem in her infant's gut. She is shaping a world. And that world, in turn, shapes us. Each one of us, every animal, all of you sitting here, developed from a single fertilized egg into a fully adult body composed of trillions of cells. And that pathway of development doesn't unfold under our own steam, under the instructions encoded within our own genome. It also occurs in partnership. It happens via conversations between us and the multitudes that we carry. So some animals don't even make that complete journey if they don't meet the right bacteria in their environment. Here's an example. These small white tubes are created by a marine worm called Hydroides elegans. Each of these tubes is very small, but they can get so dense that they can clog the propellers of ships. They can carpet the surface of underwater ship pilings and vessels. And these tubes only form when the worms reach their adult state. And the worms only reach their adult state when they encounter the right microbes in their environment. If all the bacteria in the oceans were to disappear, these worms would never be able to become adults. They would be like Peter Pan, forever trapped in childhood or in larvahood, and never be able to complete their life cycle. Within a generation, they would disappear. Many other animals that we are more familiar with, like mice, for example, rely on microbial signals throughout their entire lives to shape the development of their organs. And we can see this very clearly by looking at creatures like this mouse that has been completely disconnected from the microbial world. This animal, like many other in facilities all around the world, was raised in sterile conditions. It does not contain multitudes. It is like a silhouette that hasn't been filled in. It is one of the strangest animals on the planet as a result. One of the few that has no bacteria and has never encountered a bacterium before. And as a result of that, it has many, many physiological problems. Its bones don't develop properly, nor do its blood vessels, its gut. Various different parts of its body are imperfectly formed because it has never encountered those microbial signals. And it's testament to how microbes shape and sculpt our bodies. They help they influence the development of our organs. And that includes the immune system, the thing that's meant to keep us healthy. The thing that, according to popular belief, is like a military force that hunts and destroys things that are other and non-self, and that includes the bacteria that are around us in the world. And yet that metaphor cannot possibly be correct, because we know that actually microbes help to stimulate the growth of many parts of the immune system. They are responsible for the promotion of many kinds of immune cells, and they actually calibrate the immune system. They help to strike a right balance between inflammatory responses that will help us deal with infection, while also stopping us from overreacting to imagined and harmless threats, things like dust and pollen and other allergens in the world around us. It is thanks to microbes that we can calibrate our response, those kinds of responses, that we can strike a balance between reacting but not overreacting. And in return, the immune system selects for the microbes that live with us. Rather than indiscriminately killing them, as this metaphor would suggest, it gets to decide which species get to live with us and where they get to live. So I think of the immune system more as a team of rangers in a national park. They care for the species of the park. They decide who gets to live there, what their population sizes should be, and they chuck out invaders. And finally, microbes also influence our behaviour. There have been many studies using rodents, mice and rats, including those germ-free ones I showed you before, showing that the microbes in our guts can influence the way we behave, the way we think, everything from our resilience to stress to our susceptibility to anxiety. Aspects of personality and mood have this microbial influence. To what extent that influences human behaviour is still unclear, but it is entirely plausible that the microbiome shapes the way we think. There are many connections between the gut and the brain. There's a so-called gut-brain axis, a two-way line of communication. There are nerves that run from the gut to the brain. The immune system can send signals between the two. And bacteria in our gut produce chemicals like dopamine and serotonin, chemicals that we normally think of as responsible for signaling in the brain. So thought, immunity, development, digestion, health, all of these aspects of our lives that we think of as the province of individuals are in fact the result of partnerships, of collaborations between us and the organisms that live with us. And those partnerships matter over the course of an individual life, but they also matter profoundly over longer evolutionary timescales. Because time and again, we have seen that microbes can influence the evolution of different animal groups, allowing them to take up opportunities that would otherwise have been denied to them, to succeed often against their very fundamental animal nature. Here's a good example. These are sap-sucking bugs, an aphid, the cicada, and a leafhopper. All of these are part of a group of insects that have made a very successful living drinking the sap from trees, from plants. And they can only do this because they have bacteria living inside their very cells that provide them with nutrients that are missing from that diet of sap, nutrients that these animals cannot make on their own. Without those microbes, these insects would never have been able to make use of this plentiful food source, one of the widest in the world. And they would never have radiated into the 85,000 species that we already know of. This is an extraordinary example of bacteria fueling the success of an extraordinarily successful group of animals. There is an even better, more extreme example of this idea of supplementation in the deep ocean. These are more tube worms, but they are huge, meters in length. They're called riftia. They look a bit like tubes of lipstick, whose ends have been pushed out all the way. They live in hydrothermal vents, areas where food is very scarce or possibly absent. And they have bacteria filling up their bodies that provide them with all the nutrients they need, that use sulfides, minerals from the vent environment, to make nutrients in the same way that, say, plants on the surface would use sunlight to photosynthesize. They chemosynthesize. And through doing that, they provide the worms with everything that they need. So to the extent that these creatures have no mouths, no guts, no backsides, no nothing. They have no digestive systems, because why bother eating when you have microbes to provide you with all the sustenance you need? Of course, most animals have kept their guts, and some have expanded them to great size to act as massive fermenters, essentially hotels for microbes, for them to do their digestive work. These wildebeest are a good example. Cows, goats, sheep, all the grazing animals that you're more familiar with, these too rely on microbes for something like 70% of their total energy budget. Again, kill all the microbes on the planet through whatever means you like, and all of these would die. The vast plains of Africa would be silent and quiet. Aside from providing these kinds of evolutionary opportunities for animals, microbes can also accelerate the pace of animal evolution. And they can do that because rather than having to accrue gradual changes in our own genomes, we can adapt to new challenges by relying on microbes that already have those adaptations. So rather than, for example, filling up a skills gap in our organization by training the existing staff with those new skills, we can just hire people that already know how to do the job. That's what this insect does. This is a bean bug that lives in Japan. And it can become instantly resistant to one of the most commonly used insecticides in Japan if it swallows the right bacterium in the soil, bacteria that can detoxify that chemical and neutralize it within the bug's body. This adorable desert wood rat does much the same. It can become instantly resistant to the toxins of a plant that is incredibly common in the deserts it calls home, again, by swallowing the right microbes in the world around it. And these aphids can become resistant to a parasitic wasp that would otherwise kill them. We're using a bacteria that they can trade through sex. And this is one of the very rare cases in nature of a desirable venereal infection. These kinds of rapid adaptations are useful to humans as well. This is a type of seaweed that has been harvested from the coasts of Japan for many centuries. It's used to make nori, which is the stuff that your sushi is wrapped in. Nori is currently cooked, but it used to be eaten raw. And because of that, people would swallow marine bacteria that lived on the seaweed and were very good at digesting the unique blend of carbohydrates found in these foods. And those bacteria entered the guts of those Japanese people of long ago, and there they encountered gut bacteria. And then they did something that bacteria do all the time that is extraordinary to us. They exchanged genes. You and I can only pass genes down vertically from parent to child. But bacteria and other microbes can swap genes all the time, as easily as you or I might swap phone numbers or ideas or gifts. And that is exactly what they did in the guts of those people. The marine bacteria gave the gut bacteria genes for breaking down those carbohydrates in seaweed, which is why, to this day, Japanese people have a gut microbiome that is uniquely qualified for digesting the carbohydrates in those foods. Okay, so microbes are cool, microbes are influential. I hope you all buy that now. Here's one thing that's really important to say. Microbes are not our friends. Even those that stay in our body and do us good are not inherently our allies. They are not our buddies. This old narrative of germs that we need to destroy, of bacteria as villains, has been replaced by a newer narrative of friendly bacteria and good microbes, but both of these ideas are equally wrong. In truth, these organisms are just part of us, or in fact, we are just yet another ecosystem to them, another habitat in this world that they have lived on for billions of years. So the bacteria that live in our guts, for example, even though they may help us to digest our food, and even though they may train our immune system or maybe influence our behavior, if they cross the lining of the gut and enter our bloodstream, they can cause inflammation and sepsis. They can cause terrible illnesses. For the matter of just a millimeter, our allies can turn into our enemies. Which is why I say that there is no such thing as a good microbe or a bad microbe. These are terms that belong in children's stories, and they are ill-suited to describing the messy and contextual relationships that abound in the natural world. In reality, we all need ways of containing our multitudes, of keeping them in line. We use physical barriers to ensure that they stay in the right place. We use our immune system to keep them in check. We feed them with appropriate food so that the right species get to live with us, like the sugars found in breast milk do for B. infantis. And when those measures break down, as they sometimes and actually often do, then our partnerships with our microbes can turn sour. And you can see some of these dynamics at work in coral reefs. Corals, even though they look like massive hunks of rock, are in fact animals too. And they too have thriving microbiomes of their own. They also have competitors, turf algae, kind of scummy, seaweedy things that compete with the corals for space. And many of the things that kill corals, overfishing, nutrient pollution, and so on, do so via changing the balance of this conflict by giving the algae an advantage. And as they grow, They release dissolved sugars into the water, kind of fast food that nourishes microbes that live on the corals, and specifically, preferably, those that are likely to cause disease. So the entire coral microbiome shifts from a normal, healthy community into one that's more likely to kill them. Now, this is a very different view of disease than what we are used to. It's not, say, like tuberculosis, the work of one bacterium, or plague or leprosy, or any of those other canonical microbial diseases. This is a case where an entire community has shifted from a healthy state into a sickly one. And there's a term for this, it's called dysbiosis, like the dark mirror of symbiosis. Organisms living together for the poorer of the whole. You can see examples of this in the human body too, because there are many, many, many diseases, disorders, a long laundry list of them that have been linked to changes in the microbiome, a list that's so long that it almost beggars belief and invites parody. It's still unclear whether these connections are the work of the microbes causing the disease. This is the other way around, whether the microbes are a consequence of the disorder, whether it's both or whether it's neither. But there are some experiments that suggest some kind of causal influence, and they involve those germ-free mice that I showed you about. There have been studies, for example, where you can take sick rodents and take their microbiomes and implant them into these sterile individuals. And you can see that many of the symptoms of those conditions transfer across. This has been done for things like inflammatory bowel disease, for diabetes, for obesity and malnutrition. In many cases, moving the microbes across moves some of the symptoms of the original donor animal. And that suggests that the microbes are not just going along for the ride, to an extent they're also grabbing the wheel. To what extent they're doing so, whether this actually matters for human health out here in the wider world rather than for these rodents in their sterile isolators, is still a little unclear. But scientists are now working it out because this has tremendous promise for changing the way we think about health and sickness. They're also trying to look at why microbiomes might change in the first place. So why the human microbiome seems to have changed and in fact narrowed in diversity over the last, say, half century. There are many possible reasons. We have moved from our traditional lifestyles, from soils and countrysides and connections with animals into these very weird urban concrete jungles. We overly sanitize the world around us. We have taken the good side of hygiene to an almost absurd extreme, where we wage an incredible war on the microbes around us with antibacterial everythings. We use antibiotics, drugs that specifically kill microbes. These have been a tremendous health good. They have saved countless lives from infectious diseases. And yet, they are also unsubtle shock and awe weapons. They kill the microbes that we depend upon, as well as those that cause us ill. And our diets have changed from what they used to be. In particular, we eat a lot less plant foods and a lot less fiber here in the Western world. Fiber is important because it contains a wide range of different carbohydrates. And no single species of microbe has the cutlery set, the enzymes needed to digest all of these. And so a wide range of plant fiber nourishes a wide, diverse microbiome. And a diet that's low in fiber leads to a winnowing of that diversity. Again, we're trying to work out whether these changes have led to problems with our health. And that is an ongoing topic of research. It's very much alive. Can we do anything about this? Well, probably, or at least theoretically. I've shown you that microbiomes can change in both positive and negative ways, and very quickly at that. And that means they can change in deliberate ways, ways that we choose and design. At least that's possible in theory. In practice, it's much, much harder to alter our inner ecosystems. Consider probiotics, things like yogurts, capsules, sachets that supposedly contain beneficial bacteria that are meant to be good for us. These products seem to be good for some things like infectious diarrhea, but in general, they are medically underwhelming. There are huge health claims that surround probiotics, and yet they rarely live up to those claims. These products contain bacteria that have been grown in industrial cultures for a very long time, sometimes proprietary strains that have been heavily domesticated. They are present in these products in very low numbers, and the species that have been chosen for such things were chosen for historical reasons, because they were easy to manufacture and package, not because they are the A-listers of our bodies, because they are good at colonizing humans. So when we take these things, the bacteria in them go in and they go out. They do not establish themselves. They are like a breeze that blows between two open windows, rattling some objects along the way, but having no lasting impact. So maybe a better idea is to give a large community of microbes, species that are very good at living in the gut. And that is certainly the concept behind a very unorthodox and slightly disgusting form of medicine called the fecal transplant, which is exactly what it sounds like. Now, it is very hard to find a good image of a fecal transplant, so I've made this incredibly technical slide. It is a way of taking stool from a healthy donor and moving it into a sick recipient. I can go over that again if it wasn't entirely clear. Okay, this sounds so unorthodox, and it is. It sounds medieval. It involves things like blenders and tubing. But it is also very successful in some cases. So it is like an ecosystem transplant. It is a way of replacing an organ without a surgery, a microbial organ. I compare it to returfing a lawn that has been overrun with dandelions, with weeds. Here's a much nicer image, there you go. A nice green field. Just focus on that for a bit. In this case, the weed in question is a bacterium called Clostridium difficile, or C. diff. It causes severe, recurring, intractable cases of diarrhea, and it can be fatal. It is a terrible thing to have. Fecal transplants have been extraordinarily successful at treating cases of C. diff. In one clinical trial in the Netherlands, for example, some 27% of people, I think, who were treated with antibiotics were cured of C. diff. Whereas people who were given fecal transplants, 94% of them were cured. And the success rate was so high that they actually had to stop the trial early, because it became clear that it was totally unethical to keep on giving people the antibiotics rather than the actual poo. Given the success with C. diff, people have tried this with a lot of other diseases that have been linked to the microbiome, things like irritable bowel syndrome and inflammatory bowel disease, but with poorer results and certainly more inconsistent results. And that's probably because C. diff is a very unusual condition. It's a case where the body has been overrun, but there's almost monoculture of weeds that seems to be easy to reset. Something like inflammatory bowel diseases might present a more complex problem. And I'm sort of waffling a bit because we still don't really understand exactly why this thing works. People have tried to replicate the success of fecal transplants with pills that contain bacteria in the gut, large communities of them, very carefully chosen, but without any of the feces or the ick factor. And the most promising of these microbial fake poop pills was tried in a clinical trial recently and failed. Same thing, C. diff, it was not as successful as actual poo, which allowed me to write one of my favorite headlines of all time, which I think will never, ever be bettered. This just goes to show how hard it is to change the ecosystems that exist within us. Because this isn't a case like, say, someone with a vitamin deficiency, someone with, say, scurvy, where they need vitamin C, give them some oranges, they'll be fine. This is about shaping an entire world. It is like managing a jungle or a coral reef or a grassland or something of that order of magnitude, something that complex with so many moving parts. We need to understand how the microbes that we might want to put in our bodies interact with each other, with the microbes that live within us already, with us, their hosts, our genes, our immune systems, how they might cooperate or compete. All these variables that we still don't properly understand, because we are still at the very early stages of this science. Even though it's been 350 years since Leeuwenhoek, even though the microbiome is such a hot topic of biology, we still have many more questions than we have answers. We still don't know all the microbes that live within us. We don't know why our microbiomes are different. So mine is very different to yours and yours and yours. And all the factors that have influenced the microbiome, the one I've talked to you about, diet, antibiotics, medicines, physical traits, put them all together and you can explain maybe 10% of that variation. What explains the rest? No one knows. And if we go into the broad... Oh, wait, no, hang on. That's a different part of the talk. I'll get to that later. There are ways in which we might use the microbiome for medical means that I think might be more promising in a more short-term way. So one of them is stratification of medicine, by which I mean working out who ought to get which medicine and who should not. There's a drug called digoxin, or digoxin, I've realized I've never said that word out loud before, that's used to treat heart disease. And it doesn't work in 10% of people because there's a bacterium called Eggopella that lives in our bodies that destroys that chemical and neutralizes it, makes it inactive. And many of our drugs, from very famous ones like paracetamol to newer ones like the latest blockbuster cancer immunotherapies, work in some people and not in others, or may have toxic side effects in some people and not in others because of the microbes we carry. So maybe in the future, doctors will look at our microbiome and say, you should get this drug and you should not. Maybe it could be a way of tailoring other medicines. And maybe we can use drugs to nudge and change our microbiomes, to persuade them to do things we want them to do and to avoid doing things we don't want to do without actually messing about with the composition of those species. Here is one example. Choline and carnitine are nutrients in our diet. They're found heavily in things like red meat, and they get changed in our bodies into two successive chemicals, TMA and TMAO. And the latter of these can slow the breakdown of cholesterol and increase the risk of fatty deposits in our arteries, atherosclerosis, and therefore heart disease. So what can we do about that? Well, that step, the conversion from TMA to TMAO, is done by one of our own enzymes, one that we encode within our own genome. And you can get drugs that block that enzyme and therefore stop that from happening, which sounds great, lower risk of heart disease. The only problem is that TMA smells of rotting fish. So it's not really something you want building up in your body. The alternative is to go for that step. And that step from collagen and carnitine to TMA, that is done by bacterial enzymes. And there are drugs, like one called DMB, which can stop those enzymes from working and therefore shut that pathway down early. Great for our health, great for our heart. Now, you probably have heard of things called statins. They are drugs that are prescribed to people who have a high risk of heart disease or high cholesterol or what have you. Maybe in the future, doctors will prescribe people who have a high risk of heart disease with a statin, a drug that targets the human half of themselves, and something like that, a drug that targets the bacterial half. Again, this is still in an early stage of development. I say a quick win. Maybe this will take 5 or 10 years to actually come to pass. Science and biology in particular is full of people making grandiose claims about medical advances and being overly optimistic about how long those advances will take to actually arrive. And I don't want to lead you down that route. Because, as I said, there is so much we still don't know about the world around us. And our ignorance gets even deeper when we look beyond the human and animal-associated microbiome and out into the wider world. This diagram is a tree of life. It is a master genealogy of everything that exists, and it came out in a paper that was published a few months ago. You don't need to read any of the writing, just look at the overall shape. And in particular, look at that spray that's coming down at about 4 o'clock, that green little feather. If I zoom into that, that is you. That is you and every other animal and all plants and so on. They are at that very tip of that little twig, of that little insignificant feather in this massive tree. The rest of that is microbial. And, in fact, a lot of that, so if you look at the red dots on that image and the one in the top half, you'll see them all over the place. Those represent twigs and branches where we have never isolated or actually seen a single member of those groups. We only know they exist because someone somewhere has sequenced their DNA, has taken a swab, an environmental sample, a bit of water from the sea, from a mine, from soil, and found the DNA of new microbes within them. This is how we explore the natural world now, not by sailing around islands with nets and forceps like Darwin and Wallace did, but by going to zoos and bodies and soil and water with cotton swabs and with DNA sequences. And that whole spray on the right, that whole purple group, that was discovered, I think, a few months ago. There is a huge portion of the tree of life, a huge part of the bacterial diversity in the world around us, and no one knew it existed last decade. So we really are at the start of this exploration. As a chap called Jonathan Eisen, a microbiologist I know, says, we know almost nothing about the biology of most of the tree of life. Now, I think it is incredibly important to explore that tree, even though it deals with creatures that are so unfamiliar to us. Because in some cases, that exploration, as Leeuwenhoek did, as others have done since him, can lead to unexpected and incredible benefits. And that's what I want to finish off with today, to tell you one last story where the exploration of the microbial world has really paid off. It begins with this guy, a man called Simeon Bert Wolbach, and his partner, Marshall Hertig. In 1924, they looked inside the body of a common brown mosquito, and they found a bacterium that no one had seen before. And no one knew what this thing was. These guys certainly didn't. They didn't know if it was important, if it was common. They didn't know anything about it. It was just there. It took 12 years for Hertig to actually give the thing a name, and he named it after his friend. He called it Wolbachia. It lives inside the cells of insects. And decades after these two found this thing, and after Wolbach himself had died, people who studied insects all around the world suddenly realized that the bacterium in those insects were all the same, and that they were all this, Wolbachia. This thing lives in some 40% of species of insects and other arthropods, which are themselves the most successful and most diverse groups of animals on the planet. This is a strong contender for the most successful bacterium in the world. It is one of the greatest pandemics in the history of life. What it does varies a lot from host to host. In some cases, it is a parasite, a manipulator of sexual biology, because Wolbachia can only pass down through the female line, from mothers to daughters. Sons and males are a dead end to it, and it has many ways of dealing with them. In this animal, the blue moon butterfly, it often just kills the embryos of any males that it gets inside, so that the adult populations can have 100 females for every male. In some woodlice, it transforms males into females. In some wasps, it allows the females to reproduce asexually by cloning themselves so that they have no need for males whatsoever. In other insects, Wolbachia is a mutualist, an ally. In this blood-sucking bedbug, it provides it with B vitamins that are missing from its meals of blood. This is a red leaf, and you can see a little green bit in the corner. That is created by an insect called the spotted tentiform leaf miner, a type of moth, a caterpillar. And that insect uses Wolbachia to stop the leaf from yellowing. It holds back the progress of autumn so that it can continue to feed. But the thing that should matter to us most is that Wolbachia has amazing properties when it gets into one insect that it doesn't naturally infect. And that's this, the tiger mosquito, Aedes aegypti, the insect that spreads such illnesses as yellow fever, dengue fever, Zika, chikungunya, and many others. Australian scientists have spent 25 to 30 years getting Wolbachia inside this insect. And they have done that because when this mosquito is infected with Wolbachia, it cannot spread the viruses behind those infections for various reasons. Wolbachia seems to compete with those viruses for building blocks, for things like nucleic acids and amino acids. It seems to do something to the mosquito's immune system that stops it from transmitting those viruses. Now, Wolbachia is also extremely good at spreading through wild populations because it is so good at manipulating the sexual lives of its hosts. So, by releasing relatively small numbers of these infected individuals into a wild population, scientists can, within a few generations, ensure that all the wild mosquitoes carry Wolbachia and therefore are unable to spread important human diseases. That has been tried in a couple of suburbs in Australia. This graph represents a span of time from January to May. The grey bars represent releases of those infected mosquitoes in the first parts of those months, and the green line going from 0 to January to almost 100% at the end of May represents the rise of Wolbachia in that wild population. that population, by the end of those months, is one that is unable to transmit things like dengue. And this approach has now been tested in big cities around the world, in places where these diseases are really prevalent, in Indonesia, in Vietnam, Australia, Brazil, Colombia. And it has tremendous potential for controlling a lot of mosquito-borne illnesses. It has interest from the World Health Organization. It has backing from the Bill and Melinda Gates Foundation. And it is interesting because of many reasons. It is relatively cheap because, unlike, say, insecticides, which need to be continuously resprayed, once you release these insects, you're off to the races. They are self-sustaining. They are also non-toxic. Wolbachia doesn't infect us, humans. It doesn't infect many of the other animals in the world around us. It only influences mosquito biology, and it doesn't even kill them. So it doesn't have any lasting ecological consequences, unlike, say, something like insecticides. Could Marshall and Hurtig have ever anticipated that this would be where their random discovery would lead to? I don't think so. In fact, as I said, Wolbach died before he knew that his name had been grafted on to one of the greatest pandemics in the history of life, into this thing that could potentially save millions of lives around the world. And I think the story of Wolbachia is the story of animal-associated microbes in a nutshell. For the longest time, we ignored these things, we neglected them, and then we feared them. And I think it's now time to appreciate them, because our understanding of the world around us is deeply impoverished if we don't. And all of this, all of the work on Wolbachia, Hurtig and Wolbach's discovery, Leeuwenhoek's discovery of the microbiome world at the start, all of that comes down to curiosity, a desire to learn more about the world around us and the things that we share it with, and the things that we share our bodies with. And it's that curiosity that led to all of the things that I've talked to you about today, and that I hope I Contain Multitudes will instill in readers in the future. Thank you for listening.